episode of Bioconversations Demystifying Science. As we continue our journey through the biologics process development, we will today be talking about formulation science. It is the process by which proteins that are purified are converted into stable, effective formulations which can be injected into patients. Joining us today is Dr. Karthik Ramani, who is the MSAT head, which is Manufacturing Science and Technology head, currently at Biocon Biologics. Karthik got his PhD from State University of New York, Buffalo, and following that PhD, he joined Biocon in 2005. Karthik has since taken up expanded roles in various capacities, heading both process and analytical sciences function. He's been somebody who has been involved in multiple approvals, biologics process development activities, and currently he heads the manufacturing sciences and technology function. Over to you, Karthik.
Hi, Kavita. Glad to be here and looking forward for our Bioconversations.
True. So, Karthik, you've spent nearly 2 decades at Biocon post your PhD, and it's been a shift from Buffalo, which is sunny State University of New York, Buffalo, to Bangalore. How has that experience been?
So I transitioned from actually being a graduate student into the professional world, and it was, I would say, a big eye-opener in many ways. As a student, you focus primarily on very specific topics. It's a narrow world where you try to solve an issue, you have a hypothesis, you try to answer those questions, and you write up your thesis.
Very structured.
You could say that. I think I think it's not something whether it is transitioning to Bangalore or anywhere to the industry. I think I don't believe the graduate education truly prepares you. Let me put it that way. I think
for industry.
For the industry, I think a lot happens, and I think you deal with some of the smartest individuals out there. So you have to be humble enough to know or acknowledge what you don't know, and I think you learn a lot of soft skills, which I don't think your graduate education really prepares you for.
So you've been a formulation scientist by training. I mean, your PhD was in formulation sciences, and here you led the formulation sciences function for some time before taking up expanded roles. As you know, formulation development doesn't always get the attention it deserves, and in bioprocess, in biotechnology sector. So what are your thoughts on those?
Well, there's a lot of truth to it, actually. I think the assumption behind formulation was you add and mix things, which is partly true. That's one of the way of looking at formulation. But I think there's a lot that goes behind formulation. There's a lot of science, it's a bit of art. And I think it's striking a delicate balance between all the components that you add, and you really need to understand what you're adding, why you're adding, how you're adding, to have an appreciation for formulation sciences. So depending on a layman view, yeah, it is mixing things. But how you mix, what you mix is equally important.
Correct. So once you have a raw protein that is, you have cultured, you've grown, you've cultured and purified, you get the drug substance in your lab. What is the first step that you do once you get the drug substance? How do you convert it into formulation that patients can actually use?
Okay, so I'll take the example of insulin, because that was probably the first product I started working on in Biocon. And so you get it as a kind of a dry powder. Let me put it that.
So are all drug substances dry powders?
Not really. I think when you look at the world of monoclonal antibodies and other therapeutic proteins, sometimes you get them in a liquid form.
Yeah.
But in the case of insulin, I think it can be relatable more to people who have been working even in the small molecule world. You get it as a kind of a dry powder. And now the question is that how do you convert it into a product which patients can use it?
So is there a recipe you follow, or?
Of course there's a recipe. As I said, there's a delicate balance in terms of basically what you add, how do you add, when do you add. So you have to convert the dry powder into something that the patient will be able to easily inject.
So what are the different materials that get added, ingredients rather, that get added along with your drug substance, which is the star ingredient as we call it. So what are the other different classes of agents that you add broadly? Is there a classification?
Yep. So broadly, we call the ingredients as excipients in the scientific language. Now, these excipients could be something like a preservative because you might want to utilize that product for multiple times. You might need to add a salt. You might need to add a buffering agent so that the pH of the product is maintained.
And why is it important to maintain the pH?
Well, I think just like every product, I think biologics also are even more susceptible to, I would say, degradation.
So degradation is one part. I mean, all molecules undergo a certain level of degradation. So is it a normal phenomena? How do we know that a degradation that a product is experiencing is going to impact the safety?
Yeah, good question. So as you rightly said, every product is going to degrade. That's inevitable. So the question is that how do you slow down that process?
Minimize the degradation process.
So we need to add excipients which are going to be offering stability, a protective environment to the star ingredient, in this case, the biological protein. And you have to ensure that they are added in precise, correct amounts.
And how do you determine these amounts?
Well, I think we do a lot of laboratory studies. You need to figure out how do you do this, how much to add, when to add, why to add. Those are all very important. Temperature at which you add. How do you maintain these excipients before you add them? Do you add excipients in a solid state or a liquid state or in a semi-solid state? So these are some of the things that a scientist in the lab is going to figure out before you bring everything together.
So this is a lot synonymous to cooking intuitively.
Absolutely, absolutely.
You have the same ingredient, you have the same dish, but different people make it different way, add it in different way that affects the final quality.
Absolutely. And that is why I go back to my previous statement where I said that adding the ingredients in the correct sequence is very important. So let's take the example of just a potato fry that we make at home. What do?
Eat it.
No, you first add oil. you first heat the pan. Then you add the oil, then you put mustard seeds so that there's a flavor that comes out of it. And then you add your potatoes. Or prior to that, you will sort it with onions and then you put the potatoes. You don't do it the other way. Meaning you add the potato, heat the tawa, add the potato and then add the oil. So that it'll be still potato, but something that is not palatable.
Not edible, correct, yeah. So when you say all these ingredients that you add, which is the excipients, we test the safety of the biologic product through our clinical trials. So for example, the class of ingredients that you add, the excipients, are they also safe for administration?
Yep, they are. So we typically deal with all of these excipients. They come under the broad category of generally regarded as safe. It's called GRAS, GRAS. And these are actually approved excipients from the regulatory agencies across the globe.
So there is a list.
There's a list which you can refer to. And it also tells you the safety profile of these excipients, meaning what is the maximum amount that you can add? Okay, is there any toxicity associated with it? And if so, which part of the body can potentially get impacted? So a formulator will have to know this universe that he or she is dealing with to ensure that they choose the right ingredient, the quantity, and how to add, when to add, so that the active product, which is going to be your therapeutic agent, is preserved.
So when we are evaluating that a particular composition, you evaluate the stability of your protein. So when you evaluate the stability of your protein, what are the steps that you actually take? I mean, how do you ensure that the product that you make is safe right from the time it leaves your manufacturing facility and lands into a hospital or into a patient's fridge? Largely, these biologics are refrigerated. And so how do you ensure that the product remains stable and safe for a patient to administer?
Yeah. So first thing is to understand that you cannot stop the product from degrading. It will degrade. Question is,
And when it degrades, it forms impurities.
yes, it does form impurities. And impurities by themselves need not be perceived as something very unsafe if it doesn't go beyond a certain level.
So there is a limit under which you
Yeah.
need to be
Mhm.
at,
Yeah.
and you study those limits in a
Yes.
clinical setting.
Not necessarily, it depends. Typically, when we develop the product, we try to understand what are the major pathways by which this product can degrade. We try to understand if I were to challenge this product with high temperature, or if I were to accidentally put it in a freezer, and all of these things do happen in real life.
So you simulate these conditions in the environment and basically subject your product to multiple stresses it will experience in real life.
Absolutely. So what happens is that we try to challenge the product, if I can call that, to actually force degrade it, trying to understand what kind of degradants are formed. And actually, incidentally, here it is just not only the main or the therapeutic protein that degrades. Sometimes even the excipients can degrade. The ingredients themselves can degrade, and those degradants from these ingredients, okay, can also impact the therapeutic protein. And that is why there's a lot of emphasis on trying to understand these interactions and truly define, okay, what is an acceptable limit of many of these impurities, whether there are any safety concerns, okay, and if there are any safety concerns, how do you control it, okay. These are all part of our product development world, okay, before a patient actually is able to self-administer the product or take this product.
So, for example, when the patient self-administers, most of these biologics, are they liquid form or are there solid dosage forms such as tablets? Those are they possible? Or biotechnology products, they tend to be a bit complex. So do you always administer them as a liquid?
Typically, yes, but you can also have them as a kind of a dry powder, which you can reconstitute it, and then again it is administered as a liquid.
With?
You will say sometimes it is reconstituted with just pure water, sterile water for injection. We call it sterile water.
So you provide this along with the medicine?
It depends. Sometimes it is off the shelf. You can buy sterile water for injection off the shelf, but in case this product has to be used in a clinical setting or in a hospital setting for an extended period of time, then you might have to add some preservatives in this water, which is something that we might either manufacture.
To inhibit bacterial growth.
So either you provide it, or again these are available off the shelf from the hospitals or pharmacies where you can buy it and reconstitute the product and then administer it. So typically, all the biologics are largely liquids, although there have been quite a bit of advances in the recent past where I think there's a lot of push in actually delivering the biologics via the oral route using like a tablet or a capsule that we are normally used to seeing.
So when we are, so for example, insulins are usually self-administered by patients at home. What are the patient-centric designs that you look at? Say, for example, when many of our chronic disease, non-communicable diseases, especially diabetes, it tends to be a bit painful when somebody has to administer it every day. So what are the patient-friendly, is there a way in which you make your product more patient-friendly? Medicines are not very friendly to be taken. Nobody wants to take them. But in the event you have to take them, what are the parameters that you look at in order to make it the least painful for the patients? Okay. Do you study those?
Yeah, we do. As part of our product development activities, I think we do beyond formulation. I think we do beyond mixing things. Because at the end of the day, we have to convert the product that a patient has to take into something that they can handle very easily. So when, say for example, let's take the example of insulin. Okay, so it was historically insulin was the product was filled in a vial, and then the patient used to take a syringe and then withdraw it from the vial and then self-administer it. And a lot of patients have needle phobia. So and especially imagine that you're doing this actually for a pediatric patient. Okay, and so over a period of time, I think many of the manufacturers, including Biocon, have converted or rather moved from vials to actually administering the product via disposable pens or a reusable pen, where it is like just like a pen like this. So the product is within this. You can actually take the product,
yourself still,
dial, and then poke. but you don't see the needle, or the needle is so fine that the patients don't have any phobia. So that eliminates that. Eliminating the needle phobia is very, very critical to ensure that patients are far more compliant in taking those drugs, because nobody likes to poke themselves.
Correct. Yeah. So, for example, there is a certain frequency at which each of these medications need to be given. So, by working on your formulation in such a way, do you make a product that essentially helps you reduce the dosage frequency?
In fact, that is one of the very key requirements for ensuring that patients are far more compliant, because nobody likes to poke themselves every day.
So, I'm sure you want to control your disease and you will have to take medications, but it will always be helpful if you have to say poke yourself once a day versus 3 times a day.
Well, I do agree, and that is also the reason why I think we try to understand what kind of frequency of dosing that we will need to plan for a patient. So, for example, for a diabetic, typically, people are going to eat every day. They're going to have a breakfast, they're going to have lunch, they're going to have dinner. So, obviously, you're going to have glucose spikes coming up for a diabetic. So, obviously, the frequency of administration will be quite high. And then over a period of time, I think many of these frequently administered insulins became also what they call it as long-acting insulin, where you take only once a day. And now we have drugs which are getting developed. In fact, I think recently, I think once a week insulin actually got approved in Europe, if not in the US. So the manufacturers and the scientific community is pushing the boundaries of the frequency of administration, okay, to ensure that it is more patient friendly.
Correct. So, what are the advances that are happening in the formulation? I mean, not in the product per se, not the molecule itself, but in the way the product is administered. What are the different dosage forms or route of administration or excipients that are getting evaluated to help improve patient experience?
Yeah. So again, I think medical device I see as an extension of basically your formulation. You have to build the device around the formulation and the product itself so that it becomes user friendly.
So, as a formulation scientist or a formulation developer, what are the key people in the ecosystem a person is expected to interact with to understand more about their product?
Okay.
Or develop a product that is one of the best?
Yeah. So, as a formulator, I think I'm very close to the patient first.
Yes.
So, I think because the product that you develop in the lab is what the patient is going to be taking. So, obviously, the patient ecosystem is very important. A doctor who is going to prescribe your formulation is also is an important key stakeholder for you. So, these are external. And then you're going to have the internal stakeholders. You're going to interact with colleagues from manufacturing. You're going to interact with colleagues from from quality control, from quality assurance, from regulatory affairs, and of course colleagues from your own R&D team. So I think it's an extremely interdisciplinary cross-functional effort. As they say, it takes a village to raise a child. To develop a product, I think different departments will have to come together to make that product successful.
So it's the width of the network, it's the breadth of the network, your own development, as well as interacting with each key stakeholder to understand how your product is.
And that is what the graduate school doesn't teach us.
Correct. So thank you so much, Karthik, for helping us demystifying the scientific part of formulation development, which is a very crucial part of a biologics product journey, because in this form, the product makes its way into the market and the patient's house. So before we end, I would like to ask you some fun questions, or just some experience questions. So you transitioned directly from academia to industry. So I know you mentioned or touched briefly upon some of those aspects, but what are the couple of points that a student must remember before making a foray from academia into industry?
Okay.
There might be multiple, but maybe one or two that you think is a key skill that they would need in order to succeed in the industry.
I think be humble. There are smarter people than you, so you have to learn from them. And the other important is be open to learning. Be very agile. I think these two things are totally important for you to succeed in any field.
From Buffalo to Biocon, How's your experience? I mean, you were one of the founding members of the formulation lab here 19 years ago, and I'm sure that experience has been very valuable from where you started and where you are now in a leadership position. So, what has your experience been in terms of growth within the ecosystem?
I think it's been fantastic. I can't ask for anything better. Actually, when I moved back from Buffalo to Bangalore, I didn't even visit the organization when I came. I just walked in. I think all my interviews happened virtually, and I just packed my bags. At that time, I was not married, so it was easy to just pack my bags and come. And I just landed up here without any expectations. And I think that also kind of allowed me to have this flexible mindset to take things as they come by, rather than having a preconceived notion that this is how things should be. In fact, Biocon has been my first and the only company so far, and I think the journey has been simply fabulous for me.
And you've been very open to the opportunities that came through.
Absolutely. I think that is very important. One of the things goes back to your previous question is to grab whatever opportunities come your way.
So, one last question. You're a formulation scientist and we touched about cooking. So, what kind of a chef would you be? A methodical chef or somebody who is very impromptu and spontaneous?
Okay, I think I will answer this question in this way. When I start, I would like to be a math scientist. Just explore things, add things, see how things are. But once I get the formula,
you want to stick to it.
I want to stick to it because you have to scale it up, and you have to do it consistently, because that is very important. We are in the business of saving patients.
Correct.
And you cannot experiment. Okay, I always hear this, you can experiment in the lab but not experiment in manufacturing, which is absolutely true. You have to get that formula right. So there is a stage up to which you can experiment, be a mad scientist, but once you
have to freeze things.
Once you freeze things, and once you start getting it out of your lab, it has to be as consistent and reliable as possible, because there is somebody out there, a patient, who's waiting for your drug, okay, and it is a life-saving drug, and there you cannot experiment.
So that's all from this segment today. Stay tuned for our next episode in Bioconversations Demystifying Science.
Thank you, Kavita.
Thank you, Karthik.